All right, good morning. I'd like to thank you for being involved in this introduction to avian influenza. Hopefully we'll talk about some of the general things about avian influenza that are important for laboratory diagnosis, and as well as explain some, I think, what are some of the more important factors or features of avian influenza as it relates to disease control. My name is Dr. David Suarez. I'm from the Southeast Poultry Research Laboratory, where I'm the research leader for the emerging and exotic avian disease viral group that does study avian influenza and Newcastle disease. So to start out my talk, let me just tell you a little bit about avian influenza and specifically some of the important proteins that are involved in avian influenza replication as well as attachment into the cell. I think probably the two most important proteins that we have for avian influenza are the hemagglutinin and neuraminidase protein, which are the two surface glycoproteins on the outside of the virus, and these proteins are the ones that do attach to the cell and allow it to gain entry. We also know that these two proteins are very important as far as virulence is concerned, particularly the hemagglutinin. Now, one of the most important things for influenza as far as vaccination is concerned is that antibodies to the hemagglutinin and neuraminidase proteins are what are protective for avian influenza. So particularly antibodies to the hemagglutinin are critical to have a good vaccine, and so you do want to have high levels of specific antibody to the hemagglutinin that you're interested in. Now, unfortunately, the hemagglutinin and neuraminidase proteins are extremely variable. There's actually been 16 different subtypes of hemagglutinin that have been described, and we'll talk a little bit more about the differences, what actually defines a hemagglutinin in the next slide. But essentially, that creates a lot of variability, which makes it very difficult to vaccinate for avian influenza. Now, probably the other important feature about avian influenza that I want to talk about today is that it is both a negative sense RNA virus, which in part means that RNA viruses are very error prone, have a very high mutation rate, which means that these viruses adapt very quickly or have the potential to adapt very quickly. And the other feature that is often that helps with the adaptability of avian influenza viruses are the fact that it has a segmented genome. You can see that there's 8 different gene segments that encode for 10 different proteins. And the importance of these gene segments is that if you have 2 different influenza viruses that infect the same cell at the same time, that they can reassort or recombine these different gene segments and come up with a lot of progeny virus that are much different from the parent virus. And that is a very important process for how influenza viruses adapt, particularly to new host species. And I oftentimes like to think that the segmented genome that influenza has is really analogous to sex for viruses, so it allows progeny viruses to be very different from the parent viruses. Now, when we talk about influenza nomenclature, it's important to have a way to standardize how we talk about different viruses, and so that if I refer to one virus, you know exactly which virus that I'm referring to, and we do that by our influenza nomenclature. We start out by talking about what the antigenic type is, and that can either be type A, B, or C. Now, for our purposes, all avian influenza viruses are type A influenza viruses, so for the remainder of our talk, I won't include that A designation. We'll assume that all the viruses that we talk about are type A influenza viruses. Now, the type B and C influenza viruses are all human influenza viruses. And so for the purposes of our discussion, we won't talk about those for the remainder of the talk. The second thing we always talk about is the isolate or the host of origin. So in this case, this virus was isolated from a chicken. And typically we will, again, give the host species of origin. Unless it's human, then oftentimes that will be left out and just will be assumed. Then we also will include the geographic location of the virus. In this case, it was isolated in the state of Pennsylvania in the United States. Geographic location can be a city, can be a state, it can be a province, or the country itself. Fourth, we also include the isolate reference, which is basically the laboratory designation that the lab gave that isolate. So in this case, typically it was the 1,370th isolate that they had classified for that year, or the isolate number that came in. And that isolate number can be anything that the laboratory wants to designate that virus so that it is unique, so that it is not represented by any other virus isolate. And finally, we also include the year of isolation. So in this case, it was 1983. Now, the last two parts are oftentimes done but are not required. That's to include both the hemagglutinin and neuraminidase subtype of the virus. So in this case, it was identified as an H5 hemagglutinin with an N2 neuraminidase subtype. And that just allows for these viruses to easily be identified or more easily identified so that we can recognize that this was an H5N2 and not an H7N3. It just makes it easier to describe what this virus is, or it gives at least some biological information about the virus. All right, we talked about influenza subtypes. We had said that there's 16 hemagglutinin subtypes and 9 neuraminidase subtypes, and they can occur in any combination. So we can have H5N1, we can have an H5N6, we can have any of these different combinations, although some combinations seem to be preferred. For example, in the United States, H5N2 are certainly the most common subtype that we see with H5 influenza. H7N7 is another very common subtype that we see in poultry. But we do know that these subtypes can occur in any combination, and again, that can be useful for epidemiology. So I promised to tell you what defines a subtype, and it's essentially antibody to one subtype will neutralize the virus of any virus in that subtype, but will not neutralize virus from any other subtype. So antibody to H5 influenza will neutralize viruses of any H5, but will not neutralize antibodies to any other hemagglutinin subtype. So it won't do H7, it won't do H10. H5 antibody only protects against H5 influenza. And what this really is important, of course, is in our vaccines, is that we have to vaccinate. If we wanted to vaccinate for all influenza, we would have to vaccinate against 16 different hemagglutinin subtypes to try to provide protection for all the different subtypes. And that makes it much more difficult to vaccinate because of the number of different antigens that we would have to include in the vaccine. Now, we do see in some cases that we will occasionally have some cross-reactivity between different subtypes, and the ones we probably most commonly see is between the H7 and the H15 hemagglutinin subtypes, but that's relatively uncommon. And generally, if you're doing a panel of serum samples, you can very easily differentiate the different hemagglutinin subtypes based on what is the highest reactivity that you see with each subtype. Now I want to switch gears and tell you a little bit about the natural ecology of avian influenza because we really consider that avian influenza is a virus where the natural host is in wild birds. And primarily we're talking about in wild ducks, shorebirds, and gulls. We've considered that the natural host for avian influenza, and that all the other host species, although they can be infected and it can be a problem in other host species, we really consider them abnormal hosts. So for our poultry species like chicken and turkeys, we don't really think of avian influenza as being a natural infection of these bird species, but that again these viruses can get into chicken and turkeys and become can become a problem and can become endemic in those species, and I'll elaborate on that in just a few minutes. But it's also important to recognize that we really don't know what the complete host range of avian influenza is, and that it is a very promiscuous virus, and that it can obviously infect a number of different host species, including both mammalian and avian species. Now, most of the isolations that we've seen have come from the two different groups of birds, the Enceriformes and the Charadriformes, which include the ducks, geese, and swans, and the Charadriformes are the gulls, terns, plovers, and other shorebirds. However, even within these different groups, we see that certain species of birds are much more likely to be infected and are probably much more important in the propagation of these viruses over time. So with the Enceriformes, certainly we see that mallards are one of the species that are very susceptible to infection, and that certainly at certain times of year we see a very high incidence of infection. So mallards and other dabbling ducks like blue-winged teal are oftentimes infected with avian influenza viruses. And again, we've seen that we don't really know what the complete host range is. It's a very complex ecology for avian influenza, and we're still trying to understand all the different factors that are involved. Now, as far as the geographic range for avian influenza, we feel like it is a worldwide infection. Although most of the studies that have been done looking in wild birds for avian influenza have been in the northern hemisphere, in North America and Europe and Asia, we have seen isolations from other places, including Australia, Africa, and South America. And so although I think that there is a higher incidence in the northern hemisphere, the virus can be found worldwide, again, in wild birds, although at this point no isolations have been reported in Antarctica, although there have been some serologic samples from positive from penguins showing that they have been infected with avian influenza. Now, when we look at waterfowl surveys, I told you that there's 16 hemagglutinin subtypes, but we don't see these 16 hemagglutinin subtypes equally distributed in the wild bird population. So in the studies that have been done primarily in North America, we see 3 particular subtypes that are found in very high numbers every time we sample. And that's H3, H4, and H6. So if you count those 3 hemagglutinin subtypes in almost every study that's been done in North America, those 3 subtypes make up over 50% of the isolates that have been recovered during those studies. Now, we haven't really gotten into it yet, but for poultry species, chickens and turkeys, H5 and H7 are very important because H5 and H7 are associated with the highly pathogenic phenotype of avian influenza. Now, H5 and H7, however, are uncommonly found in wild birds, where typically we see in these studies less than 5% of the samples that we come back are positive for H5 or H7. So even though we see a much higher prevalence of infection in poultry, in wild birds it seems to be a very uncommon subtype. Now, we also do see big differences year to year in the distribution of hemagglutinin subtypes. So one year we may have a predominance of H6, where the next year at the same site, sampling the same birds, we may see a predominance of H3 isolates, for example, from year to year. And so actually, it's obviously a very complicated ecology for avian influenza and something that we're still trying to understand. Now, when we talk about avian influenza in poultry, I think, again, it's important to understand that certainly chickens and turkeys are not considered natural hosts for the infection, and even domestic ducks are not normally infected with avian influenza. And we feel that the main model for viruses coming into our domestic poultry species are as interaction with wild birds, so that infected wild birds will shed virus, and then that virus does infect our domestic poultry species. Now, obviously, we think there are differences in susceptibility. Obviously, domestic ducks, being closer to wild ducks, are more likely to be infected, or it's easier to infect wild or domestic ducks compared to other birds. And we even feel like turkeys are more susceptible to infection than chickens are. And so we do see these differences in the susceptibility between the different species. Now, what happens, though, is that on rare occasions we'll get a virus that does jump from wild birds to our domestic species, chickens and turkeys. The virus has an opportunity to continue to replicate and transmit bird to bird, and these viruses over time will start becoming better adapted to avian influenza, or better adapted to chickens and turkeys, so that eventually they will become adapted to those host species, and then really they have become chicken or turkey viruses, and they no longer will replicate very well in the original host, the wild waterfowl birds. And once these viruses have become adapted to chickens and turkeys, they become much more difficult to eradicate, and they're much more difficult to contain. Now let's talk a little bit about avian influenza and really differentiate between infection and disease. Infection has to be separated out, that you can have, in a lot of cases, a bird can be infected with avian influenza and not show any clinical disease. And in other cases, obviously, they can become infected and show very severe clinical disease. And we see a wide range in between. Now, I find the easiest way to differentiate or talk about infection with avian influenza is talk about the type of infection it is. It's either, we can break it down into 2 different groups. We either have a localized infection, where the virus is replicating on the mucosal surface, either in the respiratory tract, in the lungs, in the trachea, or in the enteric tract. And in that case, we consider that a low pathogenic avian influenza virus. And then we also have the systemic form of the virus, where the virus is replicating not only on the mucosal surfaces, again in the respiratory tract and in the lungs and the intestines, but it also is replicating internally in the different organs in the body. In that case, the systemic form of the infection is what we consider highly pathogenic avian influenza. So for the localized form, we may or may not see clinical disease, and typically it is either a respiratory disease or in some cases an enteric disease. With the systemic form of disease, then we can see certainly the respiratory form, but we can also see widespread infection in all the organs in the body, and that is what contributes to the very high mortality that we see with highly pathogenic avian influenza. So highly pathogenic avian influenza obviously is a very serious concern, and certainly what we're very interested in being able to diagnose rapidly. And we really define it by its ability to kill chickens in laboratory infections. So by definition, it's a systemic, rapidly fatal disease in poultry. As I mentioned previously, the high-path form of the virus is only H5 or H7. We have not seen any of the other hemagglutinin subtypes have the high-path AI phenotype. But it's also important to recognize that most H5 and H7 viruses are not highly pathogenic. Most are actually low pathogenic, and it's only just a small subset of H5s and H7 that have the high-path AI phenotype. Because the virus is such a devastating disease, it is an OIE List A disease, or what we used to call the List A disease. It's a notifiable disease. And for any country that has a high-path AI outbreak, they are supposed to report it to OIE within 24 hours of diagnosis. Now, we know that the main difference between low-path and high-path AI is actually differences at the hemagglutinin cleavage site, and I'll talk about that in a few more slides later on. And again, as I had mentioned, low-path AI, or probably one of the biggest concerns that we have, is that low-path AI can mutate to the high-path AI form of the virus. So even though we have low-path avian influenza H5 and H7 circulating in our poultry populations, we have to be very concerned about the possibility of these viruses mutating to the highly pathogenic form of the virus. Now, we're just going to show a few of the common clinical signs that we have with high-path avian influenza. But I want to start out, before showing these slides, to tell you just a little bit that a lot of these clinical symptoms are when the course of infection goes to about 4 to 5 days or a little bit longer before we start seeing the classic clinical signs of highly pathogenic avian influenza. You can have some very virulent viruses like the H5N1 viruses that we see circulating in Asia and Europe and Africa right now that actually are so virulent that they kill the birds without showing these really classic clinical disease signs. So although I'm going to show you what we consider to be common classic signs of high-path AI, in some cases you actually won't see any gross clinical lesions that will tell you immediately to be concerned about high-path AI. So in this bird, we can see here is that we actually have, both in the combs and the wattles, we can see the darkening that is evidence of hemorrhage in this case. And a lot of these signs or pictures that I'll show you are evidence where we're seeing hemorrhage in abnormal places, which shows that a lot of these high-path AI viruses are actually attacking the vascular endothelium that actually is allowing this hemorrhage to occur. So here's another case where we actually are seeing necrosis at the very tips of the comb. You can see some hemorrhage at the base of the comb that are classic symptoms. Hemorrhage in the feet is another classic lesion that we see with high-path AI. In this case, the intestines, and we can see some hemorrhagic lesions in the intestines, oftentimes associated with the lymphatic nodules in the intestines. We can see the classic petechial hemorrhages in the heart and the fat around the heart. Again, damage to the vascular endothelium that is allowing this hemorrhage to occur. Here's a case that if normally you would think this is looking at the proventriculus, where we're seeing hemorrhage at those nodules, we would classically think of this as a symptom that we would see with exotic Newcastle disease or velogenic viscerotropic Newcastle disease. But again, pointing out that just by gross clinical signs, you cannot differentiate avian influenza and viscerotropic velogenic Newcastle disease. And so in this case, the lesions are very similar. But the important point is that you at least raise your suspicion that those two diseases are likely to be causing these lesions, and further diagnostics are being done. Now, this is what I consider the standard model for how we get a high-path avian influenza virus. First, we start up in the upper left-hand corner where we have low-path H5 and H7 circulating in wild birds. From this original host or natural host for influenza, the virus gets spread to our commercial poultry, chickens or turkeys in this case, where we have low-path avian influenza virus, gets into that flock, begins to circulate and replicate, and eventually can spread to other flocks. It may be causing mild disease, may not be causing much disease at all. And then for reasons we don't quite understand, this virus will mutate to the highly pathogenic form of the virus. And when it does, obviously, that's when we start seeing the very severe disease with high mortality. So again, we feel like the virus starts in a low-path AI form in wild birds, spreads to commercial poultry, circulates for an extended period of time, and then mutates to the high-path AI form of the virus. Now, one question you should probably ask is how fast will that occur? That once it gets into commercial poultry, how fast will it mutate to the high-path AI form of the virus? And unfortunately, there's a lot of variability there, and we can't really predict how quickly that can occur. So we've seen some cases like in Canada in 2004 in their outbreak where the virus probably circulated for less than 2 weeks before it mutated to the high-path AI form. A case down in Chile in 2002, we felt the virus circulated for probably a month before it mutated to the high-path form. Our big outbreak in the U.S. in Pennsylvania in 1983, a low-path AI virus circulated for at least 6 months in commercial poultry before mutating to the high-path AI form of the virus. And then we have some cases where we have, actually in the U.S. in our live bird market system, we have H7N2 that's been circulating there for over 12 years and has not changed to the high-path AI form of the virus. So still a very unpredictable nature of disease of that emergence to high-path. Now, in the U.S. or in the Americas in the last 30 years, we've had 5 major disease outbreaks of high-path AI. And again, high-path avian influenza is considered a foreign animal disease in the Americas. It's not something we would normally find. The biggest outbreak we've had in the United States was in Pennsylvania in 83 and 84. We had to destroy, or 17 million birds died or had to be destroyed to control this outbreak. Cost the government over $60 million in 1983 dollars, which is well over $200 million today, to control this outbreak. We had another outbreak in Mexico where several million birds were involved. In this case, they were able to get rid of the high-path AI form of the virus in Mexico, but never got rid of that low-path AI form of the virus, and that's continuing to circulate in Mexico to this day. I had mentioned the outbreak in Chile. They actually had one large complex of birds that were involved, 2 million birds, but essentially the virus was contained to that one very large complex of birds. Canada was another major outbreak in 2004. Again, 17 million birds died or were destroyed to control that outbreak. So obviously a very costly outbreak in Canada. Now this last one we'll talk a little bit more about in the next presentation on diagnostics, but we actually had a small outbreak in Texas in 2004. This was unusual because the sequence of this virus suggested it was a high-path AI virus and it was reported out because of that reason, but when we actually put this virus into birds, it did not cause any mortality, and very few birds were involved in this outbreak because of rapid detection and eradication. Now, I'd already mentioned the hemagglutinin is very important. Just wanted to touch on that just briefly again. That the protein is initially made as a single large protein that has to be cleaved into the HA1 and HA2 subunits by host proteases before the virus is infectious. And we know that the cleavage of the hemagglutinin is necessary for this to occur. And this really makes the difference between whether a virus is high path or low path, is what type of proteases can cleave this cleavage site. So low path can only be cleaved by trypsin-like proteases that are found in the respiratory tract or the intestine, whereas high path AI can be cleaved by a much broader group of proteases, endogenous proteases found in almost any host cell, and that's the reason why the virus can replicate systemically. Now, the standards for high-path AI, again, I told you earlier that we define it by its ability to infect and kill poultry, chickens specifically, and that in standard pathotyping tests using specific pathogen-free birds, if the virus kills 6 or more, 75% or more of the birds within 10 days, then it is considered a highly pathogenic avian influenza virus. Now, because we know that H5 and H7 are very important for the subtypes that we need to be concerned about with high-path AI, we also will sequence the hemagglutinin cleavage site because we can, in some cases, tell that the virus has the potential to be high-path or is high-path based on the hemagglutinin cleavage site sequence. And certainly that's what we did in the Texas outbreak I had mentioned earlier. The virus had a sequence of a high-path AI virus, but biologically it did have a low-path phenotype. And then we also have to be considered that low pathogenic avian influenza, H5 or H7, is also notifiable to OIE. Although it doesn't require the 24-hour reporting, usually it has to be reported within 30 days if these viruses come out of commercial poultry. Again, I had mentioned that the main difference between low-path AI and high-path AI is the ability of these proteases to cleave the virus, and that just separates while it allows one virus to replicate just on the mucosal surfaces, whereas the high-path AI virus can grow inside the body systemically. Now, when we look at the sequence of these viruses, we see that the typical waterfowl low-path AI viruses typically have 2 basic amino acids at the cleavage site, and so that's commonly what we see. And then with all the high-path AI viruses that we do see multiple basic amino acids at the cleavage site, or insertions of amino acids at the cleavage site. So the Mexican outbreak actually has 5 of 6 of the amino acids at the cleavage site are basic amino acids. The Hong Kong 97 outbreak and all the Asian H5N1 viruses have 8 basic amino acids at the cleavage site. And in some cases, the glycosylation site can also be important, and we'll talk about, but we won't talk about that in detail today. With H7, we see really the same pattern, although there is a little bit of difference because the sequence is different between H5s and H7s. Again, we typically will see 2 basic amino acids with low-path AI waterfowl viruses. The only difference is that with high-path AI viruses, we've always seen insertions of basic amino acids, not just it's not only that you need basic amino acids, but you also need insertions of amino acids for the virus to become high-path. Now, I'd already mentioned that reassortment in gene segments is very important. Again, it's another way for the virus to increase diversity and provides one more mechanism for the virus to adapt very quickly to a new host environment. And again, this is oftentimes referred to as sex for viruses. Now, one example that we have, a classic example, is when we had the H5N1 outbreak in Hong Kong in 1997. We seem to have the virus that we had in Hong Kong appear to be actually a combination of several different viruses that had been circulating previously. Certainly we had the H5N1 viruses, the Goose Guangdong 96 virus, that was highly pathogenic for poultry, but there was no evidence that this virus could replicate or cause disease in people. And we also had an H9N2 virus that was also circulating in Hong Kong at that same period of time. We also had some H6N1 viruses, and we feel that there was a reassortment event that went on, that there was a different combination of those internal genes that resulted in a virus that was both highly pathogenic for poultry, but that was also pathogenic for humans, the Hong Kong 97 outbreak. Now, this outbreak was controlled in part because they eradicated all the poultry in Hong Kong to control this outbreak, and we have not seen these particular viruses with these exact same 8 genes since that period of time, since that eradication occurred. Now, this kind of moves us in the area of influenza host specificity. Then generally we think of influenza viruses as being host specific, so that a swine influenza virus will infect swine, but it will not infect other species of animals. But it turns out that influenza obviously has a very wide host range. It's a very promiscuous virus, and so that we do see numerous exceptions where the virus can jump the species barrier, and sometimes even the class barrier, where a virus can go directly, for example, from chickens to humans. And for example, with the swine influenza virus, we have seen swine influenza virus infections in humans, and we've also commonly seen swine influenza viruses infecting turkeys. Actually, in the United States, the one case where we vaccinate in the U.S. Is in turkey breeders, and we vaccinate those turkey breeders to protect them against infection with swine influenza virus, because if they get these swine influenza viruses, that can really hurt their egg production, which can be very, very costly for the producer. So we typically will vaccinate our turkey breeders to protect them from swine influenza. Now remember that for a host to be infected, or for a virus to become endemic in a new host population, it has to do 2 things. One, it obviously has to replicate well in that host species, but it also has to transmit well in that new host species. So if you have replication without transmission, then you're not going to have a virus that becomes endemic. And so probably the best example of that is with these Asian H5N1 viruses, where we know if they get into we know that people are susceptible to infection, and if they are infected, there's a large amount of virus that's produced. But the one thing that doesn't occur is efficient human-to-human transmission. So that's why the virus has not become pandemic in humans, is that although it may replicate well in humans, it does not transmit well. And that same principle works in any host species. The virus has to replicate and transmit well before an epidemic will occur. Now I'm going to switch gears a little bit and just talk a little bit about methods of control for avian influenza, and things that have been commonly used to try to control different outbreaks. Really, we only have 2 major control tools for avian influenza. We can use a stamping out approach, where essentially we want to identify the infected flocks and then euthanize them so that they cannot allow that transmission to occur to the next flock. The alternative to that is that we can use vaccination. Now, vaccination can be used actually in 2 different ways. It can be used as part of a stamping out program, or it can be used just as a disease control program, and I really need to differentiate between those 2 different ideas. But first, let's talk about stamping out. Stamping out can be a very effective way of controlling a disease outbreak, and it has been the way we've controlled most foreign animal diseases in the U.S., including avian influenza. Now, the problem with stamping out is it does require both good veterinary infrastructure as well as a good diagnostic network. Now, good veterinary infrastructure, because it does require a lot of manpower to go out and sample the infected farms, as well as a good laboratory diagnostic network that can take those samples and quickly identify whether the flock is positive or negative. And if it's positive, then they need to be quickly euthanized so that that flock will not be a source of infection for other flocks. This approach can be cost effective if the outbreak is identified very early. So if you only have to eliminate a few infected flocks, this can be a cost effective approach. But if the virus has been widespread in the area, then it can be very costly, and obviously that makes it, in some cases, not a practical approach. Now, one example that I like to say is actually in the area of foot-and-mouth disease. In England in 2001, when they had their FMD outbreak, the virus was widespread in their country before it was originally recognized. And although they still use stamping out as a control tool, it cost them literally billions of dollars to control FMD in their country because they had to identify and slaughter so many infected farms and so many animals. Where if they would have been able to catch the outbreak just a few weeks earlier, when it was on the original source farm, then they would have been able to control it at much less cost. Now, again, to vaccinate or not to vaccinate, what can vaccines do for you? Certainly, vaccines are very good at preventing clinical disease, but it's important to recognize that vaccines do not prevent clinical infection. So the birds can still be, they will appear to be normal, but still may be shedding virus. Good vaccines, if properly administered, can reduce virus shedding, sometimes to a great deal, from infected birds, and obviously that reduces the chance of the virus spreading to other flocks. And then also, vaccinated flocks are more resistant to infection. So a well-vaccinated flock will take more virus to infect than a non-vaccinated flock. And so that greater resistance and the reduction in virus shedding are very important tools to block the virus from transmitting from flock to flock, and that can be a very important tool for your vaccine or for your control program. Now, vaccines obviously do come at a cost. Not only the cost of administering the vaccines, but it also can have effect on your export market. So if you're a country that exports a lot of poultry, most countries will not accept poultry from countries that vaccinate. And so it can adversely affect your export markets. And in some cases, we can actually see where bad vaccines may contribute to clinical disease or contribute to virus spread, because a bad vaccine will, again, block clinical disease where the birds don't get sick, but still allows a lot of virus to be shed, where that virus, then you have lost one of your important control tools. You can't see that sick birds are evidence of virus infection, and therefore the virus, in some cases, can spread more widely than it otherwise would have. Now, I really try to emphasize that a good vaccination control program is not just the fact of going out and vaccinating the birds, but it's actually an integrated program where you have disease surveillance, where you have diagnostic tests, you have increased education, you increase your biosecurity, you have quarantines, you have animal movement controls. If you don't have all of those pieces of the puzzle, your vaccine program is not going to be effective. Again, vaccination can do a good job at reducing your susceptible population, and it can be used as part of a control program in conjunction with stamping out. But even if we identify that we have a flock of birds that has been vaccinated and then has become infected, still we're going to go ahead and eradicate that flock. We're not going to take a risk of that flock spreading the virus, and so that's where your surveillance program really becomes important. Again, vaccination without the proper controls may eliminate clinical disease, but it will not eliminate the virus unless it's done properly. Now, we just have one example of some vaccine trials that we've done at our laboratory. So in this case, you can see that in group 1, we vaccinated them with the Newcastle disease virus, and then we looked for whether the birds got sick, morbidity, as well as whether they died, mortality, and then we also looked for the amount of virus that was shed 2 days post-inoculation or post-challenge. So when we look at group 1, the control birds, All the birds got sick. All the birds died within 2 days because we challenged them with one of these H5N1 high-path AI viruses from Indonesia, which is, again, one of these very virulent viruses that we're talking about. And in those birds, from the tracheal swabs, we were able to isolate or show that these birds were shedding over 6 logs of virus in the samples that we were taking. Now, in contrast, groups 2 and 3, we vaccinated them with 2 different types of killed influenza vaccines. These are oil adjuvanted vaccines. One is a Mexican strain and one is a European strain. And both gave us very similar results, where in one case, with the birds that got the Mexican vaccine, none of the birds got sick and none of them died. In the European strain, we did have 1 bird got sick and 1 died, and which is not uncommon. Again, when you're vaccinating a large number of birds, some birds are just not going to respond for reasons we can't easily explain. But what I think is more important was when we look at the amount of virus shedding, is that the birds were only shedding 1 to 2 logs of virus. So we saw a 4-log reduction in virus shedding from the control birds to the vaccinated birds. So we're seeing 10,000 times less virus between the controls and the vaccinated birds, or the vaccinated birds to the controls. And obviously that great reduction in virus shedding is going to make it much easier to break that transmission cycle of avian influenza. And so I think vaccines can play a very important role in the control of avian influenza if done properly. Now, what vaccines do we have available? We have really only 2 types of vaccines that are available. We have a killed oil adjuvanted vaccine, and then we have a fowlpox recombinant vaccine. Again, both have their advantages and disadvantages, but if properly administered, they can provide good protection from clinical disease and reduction in virus shedding. Now, again, we talk about a good vaccine. What makes a good vaccine? I think there's really 2 important factors in a good vaccine. Obviously, the amount of antigen that you have in the vaccine, the adjuvant you use. Obviously, when administered, the bird will produce very high antibody levels. That's obviously one evidence of a good vaccine. Then we also need to be concerned about the viral strain, the seed strain we use in that vaccine is appropriate for the challenge strain that we're looking at. And that can be a little bit difficult to determine whether you have the best vaccine seed strain in your vaccine or not, but that is something that you can work with your OI reference laboratories to see if the viruses that you have in the field are well matched to the vaccines that you have available. And so in some cases you will actually need to do some vaccine testing to make sure that you are seeing a good response. But if you're going to, again, that's going to be part of that effort to have a good vaccine. And again, the vaccination does need to be part of an integrated program and shouldn't be used by itself, or you're not going to have good results. Now, this slide is not updated, but this is showing that there is a lot of legal vaccine being used in the world. Indonesia, China, Vietnam. I think there's vaccines being used in Russia. And there's a lot of illegal vaccination being used, and obviously we don't have a good idea for where that's going on. But again, they're all using these vaccines that we're talking about. But there's limitations to these vaccines, and probably the biggest limitation that we see is that they're not easily administered to large numbers of birds or require a lot of manpower, which makes it very difficult for them to use. With the oil adjuvanted vaccines, we also have issues of withdrawal periods, or at least in the U.S. These oil adjuvanted vaccines require a 42-day withdrawal period, which can limit how they're used. The primary disadvantage of the fowlpox vaccines is that you can use them at 1 day of hatch, but once the birds have gone out in the field, it's very difficult to use the fowlpox recombinant because if birds are exposed to fowlpox, then they will not be able to be vaccinated again. Again, control of high path has been through either eradication or by vaccination, and the U.S. has used eradication for high path AI in the past. We are using some limited vaccination for low path AI. And certainly we have control programs in the U.S. for H5 and H7. And again, strong veterinary infrastructure is helpful to control these outbreaks rapidly. Now, one of the things that we talk about is our veterinary infrastructure in the U.S., and I think we have, what we've been working on recently is trying to improve our laboratory infrastructure. And that we recognize in some of our previous outbreaks that we have our central veterinary laboratory in Ames, Iowa. And in this case, it's difficult to try to get samples to Ames, Iowa, get them tested, and get the results out quickly to help us in a disease outbreak situation. So it was recognized that we want to get regional laboratories that can do testing for avian influenza and other foreign animal diseases, so that we can get a faster response time and allow us to get a heads up or start eradication faster than we could have otherwise. And the system that we have in the United States is, this is actually showing the 12 core laboratories spread out through the U.S. There's actually about 40 laboratories that are licensed or regulated to run the avian influenza real-time PCR test, which is our primary screening test in the United States. So we have essentially at least one diagnostic laboratory in all the important poultry-producing states in the U.S. that can provide same-day testing for avian influenza. Now, with our system, we still have the federal system, National Veterinary Services Laboratory here in Ames, Iowa, are still the ones that oversee this regulatory network or this laboratory network. And any samples that are positive at in the regional laboratory needs to be sent to Ames, Iowa for confirmation. Ames, Iowa is the body that will report out a disease outbreak of avian influenza in the U.S. They are the ones who report it. They have the final say. But it's important, again, that these regional laboratories allow us to go out and test samples quite rapidly and allow us to improve our turnaround time, which has been very helpful for different outbreaks. Now, the last part of the talk, I'm going to just talk a little bit about Asian H5N1. I think that certainly that is what's on people's mind today, and tell you a few things about this virus that I think is interesting, and it makes it a very difficult challenge for control and eradication. Well, first, Asian H5N1 has not been around just recently. This is a virus that has been around since 1996. The first H5N1 virus that we can directly trace to the viruses that we see today was this goose Guangdong 96 virus that was causing disease mortality in geese in China. This had the same high path cleavage site that we see today. Again, this virus was highly pathogenic for chickens in the laboratory. And we've seen variants of this virus every year since then. In 1997, we had the Hong Kong outbreak where both poultry and humans were involved. We saw outbreaks in Hong Kong in 1999 where just geese were involved. We've seen viruses coming through quarantine stations in Korea from duck meat coming out of China, showing that those viruses were continuing to circulate in the region, although they weren't necessarily being reported to official bodies like OIE. Now, it is important also to say that even though the hemagglutinin gene for all these viruses were the same, the internal genes were quite different for a lot of these viruses, and so that's why we did see some differences in how these viruses behaved biologically in different host species. Now, of course, H5N1 really hit public awareness after the outbreak that started at the end of 2003, where it was first reported in South Korea, and then spread to a number of different Asian countries in the next few months. And it has spread to at least over 40 different countries, including European and African countries, in just the last year. But I want to emphasize that this virus is continuing to change, and so some of the things that this virus did in 1997, it is doing different things now, or is able to increase its host range to things we haven't seen before, and I'll talk a little bit more about that in a minute. So this is showing early in 2004 the number of countries that had reported avian influenza. And again, I want to point out that although the countries were officially reported that they had H5N1 in 2004, often there is evidence that there was earlier infection, in some cases published reports. Now, I have this slide just to show that at the end of 2005, in 2005, we saw something really different that occurred with Asian H5N1, and that the virus started to infect different species that we had not seen it infecting before. And particularly when we talk about in China, we have the red dot in the middle of the country. That's Qinghai Lake. That's when we saw the first report of a large die-off of wild birds in the bar-headed geese. And then just a few months later in Mongolia, in a very remote site in Mongolia where the interaction with poultry is very unlikely, we again saw an outbreak of disease in wild birds. And I think this is a symptom that this virus has now jumped from commercial poultry, has moved back into wild birds, and is now spreading and circulating among wild birds. And I do think that in a lot of cases we have seen migratory birds spreading the virus to other countries. But I also want to emphasize that poultry, movement of infected poultry and poultry products is still probably a larger role for moving virus, particularly within a country and potentially between countries. And so we don't want to just blame migratory birds for everything, because I think they are a symptom of the problem. They are not a cause of the problem. Now, differences in disease susceptibility. Again, all these viruses are highly pathogenic for chickens. Again, killing typically within 1 to 2 days by the IV route. Some of the nastiest viruses we've ever worked with in the laboratory as far as how quickly they kill. But one of the interesting things is when we started looking at ducks, we've seen differences in the viruses basically that we were working with in 1997 and the viruses that we work with today. We did a lot of work in our laboratory looking at, historically, high-path AI viruses infect chickens. It's a chicken disease, and these viruses, although they may infect ducks, typically did not cause any clinical disease in ducks at all. And essentially, before 2002, that was the same pattern that we'd seen with Asian H5N1, that these viruses could infect ducks, but they did not cause clinical disease. However, starting at the end of 2002, we did start seeing differences where we were starting to see increased mortality in ducks. And in our laboratory, we use a 2-week-old Muscovy duck model, so we give them the same dose, the same age, and it allows us to compare viruses over time. Again, all the viruses before 2002, infected ducks did not show any clinical disease. Starting after 2002, we were starting to see kind of increasing mortality, and now we've got viruses that can kill ducks within less than 3 days by a natural route of inoculation, showing that these viruses have really gained in their virulence. Again, it's difficult to understand with avian influenza, again, because it can change host species. It has this high mutation rate and it can adapt to host species. To understand or guess what the virus can do in a number of different host species, again, we know that it can infect wild birds. Seems to be very virulent for swans and some other geese. We don't know in all the cases what kind of mortality it can cause, but again, it can be quite variable, and the virus is continuing to change. Now, I do want to touch a little bit on the role of wild birds. Again, I think before really 2005, wild birds were really just incidental to the H5N1 outbreak. I think the virus, once it had become high path in chickens and turkeys, was a chicken and turkey disease for a number of years. And then now, just in 2005, this virus has spread into our wild bird population and has now at least become it has become endemic in at least parts of our wild bird population, which makes it a much more difficult disease to control now that it's in wild birds. I think all the different isolations of H5N1 and other species previously, in most cases you could say there's an association with domestic poultry. So we see virus spreading from domestic poultry into wild birds and not the other way around. But now we're seeing evidence that wild birds are spreading the virus to domestic poultry, and obviously, again, that makes it much more difficult to control. So the conclusions to my talk today is that one is H5N1 is endemic in a number of countries around the world, and it's going to be very difficult to get rid of this virus because it is so well adapted, it is so well entrenched. Again, the presence of the virus in wild birds makes it much more difficult to control because it's difficult to control that wild bird source of infection. Again, I think movement of infected poultry and poultry products are still probably more important in the day-to-day transmission of this virus, but we cannot discount the role that wild birds has. Again, the virus has shown its ability to change over time. It has been able to infect new species, again, wild bird species, and cause greater pathogenicity in species like ducks and geese. Obviously, again, making it a very difficult disease to control. And control in the short term is unlikely. Again, because it is so widespread, it's going to take a lot of effort internationally for this virus to be controlled. Vaccination, I think, can be a very important control tool, but it does need to be used judiciously. It needs to be used as part of an integrated program. It cannot be used by itself just for disease control, or we will not be able to get rid of this virus. I do think vaccination should be used, but it should be used as part of an eradication program and not instead of it. And finally, more international support is needed. We need to work together because this is an international problem. And certainly that's one of the things this program is trying to do, is to try to start building those bridges to allow better cooperation.